Okay, thank you. I'd like to start by thanking the organisers for inviting me to attend the meeting. It's a great honour to be taking part in the centenary celebrations. I'd also like to thank my fellow speakers for introducing this topic so well, so I'd be able to skip over some of the background. I guess the big question is, why are we concerned about mineral nutrition? And one of the reasons we are concerned is that, as we've heard previously, there are something like a third of the world's population that is anemic, and at least 50% of those cases are directly attributable to iron deficiency. And the major at-risk groups are adolescents and children and women of childbearing years. There's also prevalence of zinc deficiency, and that manifests itself specifically as stunting in children, and it's thought that there's maybe 155 million children globally who are affected. To combat this, many countries have mandatory fortification programs, particularly for iron, in flours from wheat, corn and rice. There's also fortification of cereals, flours with zinc. This is practiced in several countries, but it's largely done on a voluntary basis. In other parts of the world where wheat's a staple, crop milling is carried out domestically at small scale, so perhaps fortification programs aren't so effective, and we may need to consider more innovative strategies to ensure sustainable mineral levels in regions that need it most. I thought it'd be important to highlight that micronutrient deficiencies aren't only a concern for the developing world. It's also prevalent in the UK, and this slide shows the dietary reference values for the UK. We've got upper and lower limits for those values. And the figures in blue really identify the issue in the UK. They show the proportion of different population groups who are consuming minerals at levels below the lower reference nutrient intake. And we can say that consumption at this level is almost certainly insufficient to meet metabolic demand. And you can see for males, for iron and zinc, the adolescent group, there's something like 17 to 20% of those population groups who consume iron and zinc at levels below the LRNI on a daily basis. For females, the situation's even more serious. So again, in the adolescent group, there's 54% of females who are consuming iron below the lower reference nutrient intake, and zinc, 27% of that adolescent group also consuming at levels below the lower reference nutrient intake. So these groups are particularly at high risk of developing iron and zinc deficiency. We also need to consider where we get our iron and zinc from in the UK, and this is data from the National Diet and Nutrition Survey in the UK, which is a rolling programme measuring nutrient content of foods on an annual basis or on a 2-yearly basis. And you can see that for adults in the UK, nearly 40% of all of the iron that's ingested comes from cereals and cereal products. And about a quarter of the zinc also comes from cereals and cereal products. If you look at the adolescent group, the figures are even more startling. There's more than 50% of the iron in the diet of adolescents comes from cereals and cereal products. So we need to consider some of the factors that may influence the bioavailability of minerals from cereals. And I'm going to focus specifically on wheat, which is the main cereal consumed in the UK. And the first thing to consider is where the iron and zinc are localised in wheat. So on the left-hand side of the slide, you can see the structure of the wheat germ. There's a section being taken through that. The outer layers are the bran layers. In the middle, you have the starchy endosperm, which is used for making white flour. And sitting between those two layers, between the bran and the endosperm, is a layer of cells called the alerone cells. If you look on the right-hand side of the slide, you can see some fluorescence images that are showing where the iron and zinc is localised in the wheat. And you can see for iron, it's almost exclusively localised in those alerone cells. For zinc, there's some zinc in the alerone, but it's also highly prevalent in the embryo of the wheat germ. So why should this be a concern? Well, something like 90 to 95% of all of the bread that's consumed in the UK is white bread. And in doing that, we mill the wheat and we remove the bran layers. Now, while the alerone botanically is part of the endosperm, it actually attaches very strongly to the pericarp which is part of the bran. So when we produce white flour, we remove the cells that contain the majority of the iron and zinc. So we essentially denude the white flour of those important minerals. The next thing to consider is the form that the iron and zinc are stored in inside those alerone cells. So this is some secondary iron mass spectrometry imagery from colleague Katie Moore at the University of Manchester. And the important parts of this panel are down in the bottom left-hand side. Here you can see that the phytate, shown as the phosphorus oxide signal, is localized in very distinct granules inside those alerone cells. And if you look at the iron oxide signal adjacent to that, you can see that it overlays very nicely with those phytate or high phosphate containing regions. So we think that the main storage form of iron, and indeed zinc in the alerone cells, is stored in phytate complexes. And these are stored in very dense particles called phytine globoids, and these are thought to be highly resistant to digestion. The final thing to consider is the structure of the plant material itself. So we have the plant cell walls that are highly enriched in arabinoxylans, and it's never too early in the morning to show the first slide containing pictures of feces. So here I've shown an experiment in which we fed some of our flour to mice. So in the left-hand panel you can see some of the flour that we fed. The alerone cells are stained blue to show the iron, and the alerone is attached to some of the endosperm. When we feed that to mice, we can collect their feces, and you can see in the middle panel that some of those cells remain intact. So these plant cell walls are highly resistant to digestion in the mammalian gut. Now many people would say, well, is this just a problem of the mouse gut? So in the right-hand panel, this is some work from my colleague Peter Ellis at King's College London, in which they've fed a coarse wheat porridge to healthy volunteers who have an indwelling ileostomy. And this is from the ileostomy effluent. And what you can see here is very nice structure of the endosperm, the alerone cells, the outer bran layers. So again, those plant cell walls aren't broken down in the human small intestine. And what this means is if we can't break down those cell walls that contain most of the iron and zinc, then we're not going to absorb those minerals from the cereal flour. So the approach we've taken to look to see if we can increase the micronutrient bioavailability from cereals is to look at ways of processing the flour. So we've looked at a method by which we can reduce the particle size of wheat flour through a process known as micromilling. And this gives me an opportunity to acknowledge the contribution of my colleagues on this project. It's funded by the BBSRC in the UK, and my collaborators on this are Yemisi Latundidada, Peter Ellis, Sarah Berry, who are all academic staff at King's College London, and Mohammed Aslam, who's the postdoc on the project, who's done most of the work. So our collaborators, Beulah in Switzerland, they've provided us with 4 types of flour, 2 whole wheat flours, and 2 purified alerone flours, and they've been able to provide that in either a standard milled format, in which the particle size is about 100 micrometers, and also in a micromilled format, in which we've reduced the particle size down to 10 to 20 microns. The important feature here is to notice that when we do that additional processing step, we don't lose any of the mineral. So actually you can see that the iron content, the zinc content, and the phytate content don't change in those micromilled flours. What this also highlights is in the micromilled flours that we enrich for iron, zinc, and of course phytic acid as well. So we're increasing the amount of the minerals, but we're also increasing the amount of the main inhibitory component of absorption of iron and zinc in those flours. So in terms of the structure, I thought it'd be useful just to show you this. On the left-hand side, this is the standard particle-sized flour, and you can see the intact alerone cells with some of the other plant material. When we micromill that flour, you can see there's complete disruption of that cellular structure. So potentially, the iron and zinc that are contained within those alerone cells are now being released and could be more available for absorption. So that's what we've gone on to test. How do we actually look at that? Well, we've used primarily an in vitro digestion methodology in which we can make products from the flour and we can subject it to peptic digestion at pH 2 to simulate gastric digestion phase. We can then raise the pH and we add a mixture of pancreatic enzymes and bile, and that simulates the intestinal digestion phase. And then we take that digested material, spin down the undigested product, we collect the supernatant, and we can measure iron and zinc release using ICP optical emission spectrometry. And we also take some of that supernatant and add it to a human intestinal cell line called KCO2 cells, and we can measure iron and zinc uptake from those digested flour products using ICP mass spectrometry. I'll share with you a little bit of the data that we have from those studies. So first of all, look at the effect of reducing the particle size. So this is comparing the standard milled and the micro milled flour. On the left-hand side, looking at the uptake of iron, and on the right-hand side, looking at the uptake of zinc from those flours. And what you can see is just by reducing the particle size, we see a significant increase in the absorption of iron from the micro-milled flour. We don't see the same thing for zinc. So this seems to be a specific effect on iron absorption. If we now bake the flour, if we make bread from the flour, we can look again to see what might be the effect of either just having a flat bread that doesn't contain yeast, or adding yeast to the base product, baked product, and making effectively a bread roll. Well, you can see this time for both iron and zinc, there's a significant increase in the amount of iron and zinc that's absorbed when we add yeast to the bread flour mix. And we think the reason for this is that the yeast obviously contains phytase activity, and it's reducing the phytic acid content. So we've gone on to measure the phytic acid levels in those wheat flour products, and we're comparing in the left-hand set of bars the raw flour, looking at the standard and micro-milled flour. I think an interesting feature of this is that we don't reduce the phytic acid content just by micro-milling. When we bake the bread without yeast, we reduce the phytic acid content by about 50%. And we see a further 30 to 50% reduction in the phytic acid content of the flour when we add yeast. So we're getting further reduction. And we think that this decrease in the phytic acid content is what's leading directly to the increase in iron absorption that we see. Okay, to summarize the first part then of this talk, then we know that cereals are a major source of minerals and micronutrients. The availability for absorption of iron and zinc is low in cereal flours, and this is primarily due to the presence of high levels of phytate, which is the major inhibitor of iron and zinc absorption, and also the physical encapsulation of these minerals within the plant cell walls that are resistant to digestion. We've shown that if we reduce the particle size of those flours, then we increase the iron availability, but not the zinc availability in our in vitro studies. But if we reduce the phytic acid level by adding yeast to the flours, then we increase iron and zinc availability from those flour products. I just want to briefly mention another project that we're carrying out in the lab. This time it's taking a genetic approach to trying to increase iron in cereals. And this is a collaboration with Janneke Bolk at the John Innes Centre in Norwich, Peter Shuri, who many of you will probably know from Rothamsted Institute, and Katie Moore from the University of Manchester. Again, the project's funded by the BBSRC. What we've done in this method is we've overexpressed a wheat vacuolar ion transporter. And I noticed on Professor Dhaliwal's slides that he was talking about one of the candidates was VIT1, vacuolar ion transporter 1. This time we focused on VIT2. And what we've shown is if we increase the copy number of this vacuolar ion transporter, then you start to see an increase in iron content in the wheat. But you'll notice from the stained images on the left-hand side that that iron isn't just concentrated in the aleurone cell, it also spills over into the endosperm. And in the upper panel on the right-hand side, you can see that the iron is basically being concentrated in the white flour. So those are the pale grey bars. As you increase the copy number of this iron transporter, then the iron content in the endosperm increases. The dashed line across there, that indicates the level at which we fortify white flour in the UK, at 16.5 milligrams per kilogram of flour. And you can see when we increase the copy number to around about 20, we actually exceed that fortificant level in the genetically modified wheat variety. So potentially this could reduce the requirement to fortify with elemental iron in the UK. What does this mean in terms of iron absorption? Well, we've measured the baseline iron levels in the wheat flour, and we see something like a threefold increase in the wheat flour line that overexpresses the VIT2 transporter. If we look at the amount of iron released following in vitro digestion, then they're fairly similar, slightly less from the modified flour. But actually, if we look at iron uptake, you can see there's something like a twofold increase in the amount of iron that's taken up from this flour. So potentially the iron that we're getting into this transporter could be bioavailable and may be a useful addition for increasing iron uptake from different wheat flours. So to summarise the points from this final part of the study then, overexpression of a weak vacuolar iron transporter, Triticum obestum VIT2, increases iron levels in the endosperm, and therefore it enriches iron in the white flour. And the iron availability of from that VIT2 enriched white flour is increased compared to the control and standard flours. Thank you very much for your attention.
So you get an additional bonus because you have finished before time. So I open the floor for discussion.
It was the results are very interesting in terms of micro milling actually increases, I don't know, the bioavailability, or it's only the solubilization of iron and the phytate complex?
I like to call it availability because at the moment these are all only in vitro experiments. So the next plan is to take these bread products and to feed them to human volunteers to see whether that is truly an increase in bioavailability of the flour, of the iron from the flour.
I just wanted to know how is it's getting released, the iron from the complex at neutral pH or duodenal pH. Normally it gets complexed at that pH.
So we think that through the release of the iron from the plant cell walls, and then through baking, phytate is fairly heat labile. So when you bake those products, you'll reduce the amount of phytic acid, but you won't change the amount of iron that's present. So the iron phytate molar ratio will change, and that should improve the bioavailability of the iron from those flours.
My question is, how is it getting released from the complex for its final absorption at the absorptive surface? How is it getting into the Fe2 form?
Yes, so we think that the iron is taken up through the traditional DMT1 mediated transport pathway. We don't know how the iron's released from the phytate complex in the intestine, but it seems to deliver, there's some soluble iron available for absorption. But it obviously much lower than it would be from other compounds that contain lots of vitamin C, for example.
Hi. I think you partially answered my question, but still let me make it elaborate. The aileron layer is rich in many nutrients, particularly micronutrients. The undigestibility is a problem for all the micronutrients, or this encapsulation is specific for iron?
So the aileron layer is enriched in iron, zinc, lots of the B vitamins, particularly folates, and there seems to be an issue with absorption of all of them unless you can do something to break down those aileron cell walls.
My question is on the practicality of micromilling. Is this a thing that you can just do in the lab with an expensive piece of equipment, or are there cheap parts that you can swap out across India flour mills?
So in terms of the method that we've used, we're using a standard roller milling technique, and the collaborators who provide the flour, they tell me that the way that they micro mill it is that instead of having the two rollers running at the same speed, they run them at different speeds, and that can give a finer grind. But I was particularly struck by the talk yesterday, I think, of Professor Prakash, who was talking about including the chakki in the milling process in India. And it would be really interesting to see what that does to the particle size of the flour, and whether that might burst open more of the aleurone cell walls, and therefore the iron and zinc, etc., may be more available.
Isn't it possible to add phytase to the flour and to liberate zinc and iron?
It's possible to do that, but first of all you need to break open the aleurone cells for the enzyme to penetrate to break down the phytate. It's possible also to break down the cell walls enzymatically, so you can use xylanases, for example, that will punch holes in the aleurone cells. So potentially a combination of xylanases and phytases may reduce the phytate content and may also increase the permeability of the aleurone cells to release more of the iron in a bioavailable form.
Aleurone layer is the living layer. In the endosperm nucleus is also triploid, but it's living. And you put it, for making dough. De novo, it could release hundreds of hydrolases, proteases, lipases, everything. So it could be self-degraded, I think that's the message we should really convey to the audience.
Yeah.
It's a living layer. It gets automatically degraded on making the dough, and we should try to, use this particular phenomena which is there in the aleurone.
So yes, you're absolutely right. There are lots of endogenous enzymes present in the wheat that can break down to some extent the components. Yeah, and of course in Europe there's a strong tradition of having sourdough breads where you add a starter culture to the bread before you bake it, and that also has been shown to break down the phytate content to almost zero and increase the iron availability.
Is there any other question from the audience? No? I think we have had a very nice panel with very different aspects of how to increase mineral content in our mineral intake in our diet through either breeding to put more into the food, or to use biodiversity, or to use biofortification, or when we look at fortification, and then of what we need to take care of when we do fortify in order to be more effective, and the last one to have a very different approach, so that what is in the food, so that we can use it much more efficiently, so that we can, to increase the minerals in our diets. So I think we have had some very nice aspects of how to treat this one problem through different things. So if there is one or two comments or questions on these aspects of, I would like to open the discussion for 5 minutes, because we still have 5 minutes left. So if there would be any discussion about these different ranges that we have, so what would be your opinion?